So, Sushil sir, should I start now? Abhishek?
Yes, sir.
So, right now we should start, and in the inception, I would like to welcome our respected Vice Chancellor, sir, Professor Mahend Pandey, sir, Registrar Dr. Sanjeev Agrawal, Dean Pharmacy Dr. Navneet Verma, Director School of Pharmaceutical Sciences Dr. Sushil Kumar, and our resource person Dr. Abhishek Sharma, Head of the Pharmacy Department Dr. Kavita Gehlot, faculty members and students. On behalf of Fraternity of Pharmacy and IFTM University Alumni Association, I would like to welcome you all in this webinar. Today, it's matter of prestige for us that we have gathered over here to learn about a pharmacokinetic and pharmacodynamic model to understand the antinociceptive effects of mitragynine. So this activity is an integral part of IFTM University Alumni Association activity. The aim of alumni activities at IFTM University under the bannership of IUA, formerly called as IFTM University Alumni Association, is to develop and foster the relationships with our former students. Alumni means a lot to our organization, and they act as role model and provide inspiration to current students, but they also are our friends and who give us new perspectives on the education, research, and collaborations. Alumni also contribute to our global image of IFTM University, which benefits both incoming and outgoing students, and also provides the opportunities for new interesting research collaborations. So when former IFTM University and IFTM students are active in the world, this raises our profile and can thus affect the aspects such as IFTM University ranking in the list of higher educational institutes. In continuation, I would like to invite Director School of Pharmaceutical Sciences Dr. Sushil Kumar for his valuable thoughts and words of wisdom. Over to you, sir.
Thank you, Aranji, for recognition of our Abhishek ji. And really, I am feeling very well after a contact with the Abhishek ji since a long time. So, very good morning as per Indian time, and I think good evening according to Abhishek ji. So, firstly, I would like to welcome our Honorable Vice Chancellor, Professor Mahend Pandey, sir, for his valuable support and guidance for the conduction of this international webinar. I would like to welcome our respected Registrar, sir, Professor Sanjeev Agrawal, sir, for their support to organizing this international webinars. I would like to welcome and thanks to Professor Anuj Srivastav, sir, Alumni President of IFT University, for their kind support regarding the organizing of this event. And now I would like to welcome our Dr. Abhishek Sharma for their presence, and happy to see you after a long time. We are connect by the social linking. And thanks a lot for sharing your valuable experiences during the international webinar on PKD model to understand the antinociceptive effects of mitragynine and their metabolites. Really, I am very thankful to Abhishek ji for sharing with us during the event. And I would like to express my welcome to Dean Faculty of Pharmacy, Professor Navneet Verma, sir, and Professor Arun Kumar Mishra ji, and all the members of organizing teams and students for their presence and listening your views during the sharing of experience with our students. So really, I am very thankful to Abhishek ji, and thank you to all. And over to Arun sir.
Thank you, sir. And I would like to add one thing. We are fortunate enough that Dr. Abhishek has given consent for his valuable time to share his research experiences in view of pharmacokinetic and pharmacodynamic model. So, before start, I would like to introduce about Dr. Abhishek by sharing his brief profile. Abhishek Sharma is an Assistant Director of Translational Drug Development Core and Research Assistant Professor at University of Florida, Gainesville. He received his B Pharm from Mohanlal Sukhadia University, Udaipur in 2006 and became a registered pharmacist in India. He received his M Pharm in pharmaceutical chemistry from IFTM under UP Technical University, Lucknow, with MHRD postgraduate fellowship in the batch 2006 to 8 by All India Council for Technical Education. He earned his PhD in pharmacokinetics and metabolism from CSIR, Central Drug Research Institute, Lucknow. He was awarded Senior Research Fellowship during his graduate studies in 2011 to 2015 by Indian Council of Medical Research, that is called as ICMR. After his graduate studies, Dr. Sharma did his postdoctoral research under the supervision of Dr. Boni A. Avery at University of Mississippi in 2015 to 2017 and from University of Florida in 2017 to 19. So I would like to welcome Dr. Abhishek for his think tank and experiences of his research. So Dr. Abhishek, now over to you.
Thank you, Dr. Mishra. Thank you, Sushil sir. And I'm really happy to be here and available anytime whenever my alma mater needs me. And it really feels odd when Sushil sir called me Abhishek ji. So please, I'm still Abhishek, and I would love to be Abhishek like that. So I'm going to present our current research topic. We recently published it, so I thought let's talk about it. But before, I'm really happy to be here for this presentation. I have very good memories during my master's M.Pharm at staying in Asha Kaushik Hostel, and pretty good mass and a very good environment. I really miss my life, and you can see these pictures. Like, I just found some to revitalize my memories, and this is how like the current status. So apart from this, now I will talk more about science. It's a little bit emotional moment to see people after a long time. I graduated in 2008, and I think I came last in 2012 before I was going from Haridwar, where I was doing a job, and then moving to CDI Lucknow, and I stayed in between to collect my degrees and all that stuff. So as I told about the title, title is related to the mitragynine, but before going to the talk, very specifically, I would like to introduce what is mitragynine, from where is it coming, and why are we studying it. So mitragynine is a major alkaloid from a plant, crotum. Chemical name is Mitragynina speciosa, and family is in Rubiaceae, like coffee family. So this plant is not native to the United States, but it is native to, I would just open my pointer option. So this plant is native to Southeast Asia, Malaysia, Thailand, and Papua Guiana. And like you have heard of like about the opioid epidemic, and this plant has some role in it. But before going to the study, I would like to also talk about a little bit like what is happening at the natives. So in Malaysia, what happened when the Chinese moved to Asia, Thailand, in that area, they used to take morphine, and they were missing their morphine in that particular region. So the Chinese figured it out, okay, this plant can solve their morphine carving, and it can increase their physical endurance when they are working in the farms. And then the special focus like Indonesia, Malaysia, these are Muslim countries where alcohol is forbidden by the religion. So these people use croton tea. So what they usually do, they boil the leaves of this particular plant and then drink it for their social gathering, for their hangout. Now, what is happening in the United States? So in the US, people are also using it to treat their pain and opioid addiction, but there are lots of problems associated with it in terms of like adulteration, and in terms of overdoses and drug-drug interactions. So we got 2 grants from National Institute of Drug Abuse from the NIH, and we are studying this plant very systemically, and this is the part of that project. So croton, mitragynine speciosa, has more than 40 alkaloids, and today we are studying other alkaloids too. But today I will talk about the mitragynine, which is a major croton alkaloid. It is a partial opioid agonist, and it has alpha-2 adrenergic activity. It also acts through dopamine receptor. So basically, it has all those things all those pharmacological properties which are required to treat opioid addiction. So to treat opioid addiction, what we need, either we need a partial opioid agonist or antagonist, basically, which can bind to those receptors, either precipitate or subsidize that carving. And the same thing with the clonidine, like alpha-2 antagonist, it has like similar property. Now the question arises, okay, if mitragynine is too good, then why are you talking about 7-hydroxymitragynine? So 7-hydroxymitragynine is a full agonist and cause all those problems which are associated with the morphine. So it causes respiratory depression, constipation, physical dependence, and tolerance and cross tolerance to morphine. So in a very basic term, this particular alkaloid can cause all those problems which other opioids have. So if this alkaloid is a part of the plant, then that plant has to be banned, basically. And the FDA, Food and Drug Administration here, DEA, Drug Enforcement Agency, which take care of the narcotic and psychotropic substances, they tried to ban this particular plant. But people who were using it, they just raised concern against it, and they have to stop for the systemic study. If you look at the chemical structure, so this is mitragynine, and the only difference is this hydroxyl group at position 7. So I will come back and then we will talk about like what are the possibilities. So we know cotton plant, we can grow it in the US, especially in the Florida region, because it has the similar environment. And then if this plant has 7-hydroxymitragynine too, which can cause all those problems, it can treat the pain, but it can cause all those problems which other opioid can do. In that case, we have to ban it because we don't need another morphine. So we will check what are the other possibilities. So one possibility is the chemical degradation of mitragynine to 7-hydroxymitragynine. Yeah, we tested it and we can see it. If you put any oxidizing agent, KMNO4 or hydrogen peroxide, mitragynine converts to 7-hydroxymitragynine. And that's how few of the bad players in the market are doing with their croton product to make it more potent, and people will come back and buy that particular product too. The second question was, okay, is the 7-hydroxymitragynine part of the croton leaves itself? So we don't think so. We analyzed more than thousands of samples, fresh croton tea samples. We have not seen any 7-hydroxymitragynine, but we have seen 7-hydroxymitragynine in lots of finished products. So we think this problem causing alkaloid can be part of the which can formulation, manufacturing, packaging, or on the shelves too. The second problem we find out is that mitragynine converts to 7-hydroxymitragynine and CYP3A4 in liver and intestine. So again, this is the next question. Okay, if your mitragynine is converting to a metabolite, which is a problem causing, then what should we do? And this thing has already been published, available in the literature. But if mitragynine's activity is coming from 7-hydroxymitragynine, then mitragynine should have all these issues which we are, can you hear me? Which are associated with which are associated with the 7-hydroxymitragynine. And there is one more hypothesis in the literature which says 7-hydroxymitragynine back metabolizes to mitragynine. So we will try to answer at least 1, 2, and 3 of these questions using our studies. So we started, first of all, we did metabolite identification studies. So we took mitragynine and put it in human liver microsomes and in human hepatocytes and look for the different metabolites. Okay, what are its metabolites? And confirmed, yes, 7-hydroxymitragynine is a potential metabolite of mitragynine. We also looked for the other metabolites which has exposure more than 10%, because it is also required FDA to study all those metabolites if you see exposure more than 10%. And we found one more metabolite formed from 7-hydroxymitragynine, mitragynine pseudoindoxyl, which is even more potent than 7-hydroxymitragynine. So in my further slides, we will discuss only 1, 2, 3, and 4 of these metabolites, because either these metabolites are potent, binding to the opioid receptor, showing full agonist activity, and responsible for all the toxicity associated with their other opioids too. So before going to further, I can tell, so 7-hydroxymitragynine is 22-fold more potent than mitragynine. Mitragynine pseudoindoxyl is 120-fold more potent than mitragynine. 9-hydroxychorinanthidine is just, we remove this CH3 group from the mitragynine, OD methylation. Then we get 9-hydroxychorinanthidine. And here we have mitragynine carboxylic acid with the removal of this CH3, with the hydrolysis through the ester linkage. And this mitragynine acid has not been studied extensively, but according to our findings, it's potentially inactive. So the major concern, 7-hydroxy pseudoindoxyl. So before going further, we need to select one particular animal model. Okay, which animal model will be the best to study the conversion of mitragynine to 7-hydroxymitragynine and corresponding pharmacodynamic activity. So we did the same experiment, metabolite studies in human, rat, mouse, dog, and monkey hepatocytes. And we selected mouse model on the basis of the metabolism, because we can see maximum of abundance of the metabolite. The maximum abundant metabolite was 7-hydroxymitragynine. It was present in other species too, like in humans too, but this is the major, and it will be the most suitable animal model for us in terms of isolating the activity of 7-hydroxy, the contribution of 7-hydroxymitragynine from the mitragynine's activity. So first of all, we did a pilot study. So we gave mouse the same model which we identified the best for us. We gave a 20 milligram per kilogram oral mitragynine and look for all the metabolites. Like all the conventional pharmacokinetic studies, we gave oral and IV, we took pre-dose sample, and blood was collected at different time points, and plasma samples were analyzed for mitragynine as well as other metabolites using UPLC MSMS method. So what we learned from this study, we learned mitragynine converts to 7-hydroxymitragynine around 12.8% oral and 5.2% IV. Now, for those who are interested, like why are you having more oral exposure? Then answer of this problem is because mitragynine to 7-hydroxy conversion is mediated by CYP3A4, and it is all expressed in intestine. So apart from liver, when you are giving oral, you are adding the intestinal metabolism part here too. But we still have that question, okay, it is converting, but is this sufficient enough to cause something? So we did systemic PKPD study. So I'm starting the title of my presentation here after making this. So what we did, first of all, we did pharmacodynamic studies. So pharmacodynamic studies is the Hotplate antinociception activity. So if we want to know about that model, so we will take mouse, we dose the compound, take the baseline value and put it on a hot plate. And we will wait till animal will chew its leg, took it out, or jumping there. So it is an indication that animal is started feeling pain. If they can stay longer, then we cannot go more than 50 seconds because it's against animal ethics. So that's we call like maximum possible effect. And we selected ED50 dose on the basis of these experiments and did pharmacokinetic studies. And we did these studies for both mitragynine individually and 7-hydroxy individually. But when we did mitragynine individually, we also quantified its metabolite to calculate the overall exposure. So first of all, we will see the PD data, which was hot plate latency data, and we achieved 50% MPE around 165 milligram per kilogram oral dose of mitragynine and around 50 milligram per kilogram dose of 7-hydroxymitragynine. Can you please mute? I don't know. We selected these doses and performed the pharmacokinetic studies, and we give animals mitragynine, but we can see 7-hydroxymitragynine as well as mitragynine pseudoindoxil. But mitragynine pseudoindoxil concentrations was not good enough to do anything. The overall conversion of 7-hydroxymitragynine to mitragynine was 3.3%. So now you can ask question, okay, your target site is brain, but you are talking about the plasma, then how relevant is it? We are not, what is happening in the brain? So we also quantified both mitragynine and 7-hydroxymitragynine in brain. And what we observed about the brain to plasma ratio, so the mitragynine's brain to plasma ratio was 2.4%. So it means mitragynine is going more into the brain than in the systemic circulation, and which makes sense because mitragynine is a lipophilic compound and it can cross the blood-brain barrier and will stay there. Now about the 7-hydroxymitragynine, which is comparatively polar than mitragynine because it has one additional hydroxy group. It can also cross the blood-brain barrier, but it was limited and lower than mitragynine, around 1.7%. And the interesting part about it is that when you look for the brain to plasma ratio of 7-hydroxymitragynine formed from the mitragynine, it is 1.6%. But when you look at when we dose 7-hydroxymitragynine, that number is different, and it raises lots of concern. Those who are interested in gender-related pharmacokinetic studies, okay, what about male versus female? So here is the separate data. What we observed, male brain has higher exposure than females, and but for the 7-hydroxymitragynine exposure, we have more in female plasma. So we observed a sex-related differences for the mitragynine in male versus female mouse, and that those results were also correlating with our pharmacodynamic studies. So when we gave animals, so earlier data was when we gave mitragynine only and look for the mitragynine and 7-hydroxy. Now the question, okay, what will happen if you give 7-hydroxy itself? So when we gave 7-hydroxy itself, we saw 7-hydroxy as well as mitragynine pseudoindoxal. So total conversion for pseudoindoxal to 7-hydroxy was around 1%. If you remember, I started and that hypothesis was not true. The thing reported in the literature didn't reproduce, and we have not seen any mitragynine in 7-hydroxymitragynine dosed mouse. Now the same question, what about the target site? So target site wise, this time we have lower exposure in the brain. So what is happening? So there are three possibilities with the higher brain to plasma ratio of 7-hydroxymitragynine when dosed from the mitragynine. So one, we know 7-hydroxy has limited brain ability to cross the blood-brain barrier, but at the same time, we are having a pretty good number when it is formed from the mitragynine. So one possibility, mitragynine is metabolizing itself in the brain. So mitragynine can easily cross the blood-brain barrier, and then it is metabolizing in the brain, and then 7-hydroxy is staying there. Second hypothesis is mitragynine is inhibiting some sort of active transporters which are cleaning out 7-hydroxymitragynine from the brain. And in presence of mitragynine, those transporters were inhibited, and 7-hydroxymitragynine can stay longer there. And third hypothesis is mitragynine is facilitating the blood-brain barrier permeability of the 7-hydroxymitragynine. And we have tested these, we have performed these brain to plasma studies at multiple doses, different species, and we observed the similar results. So both have something going on. So the question is, okay, mitragynine is converting to 7-hydroxymitragynine, but is it really responsible for the pharmacodynamic activity? That's what we are trying to understand here. And that's why we need the PKPD model. So we have pharmacokinetic data, concentration versus concentration time profile. We do have pharmacodynamic data where we have multiple dose versus the Hotplate antinociception studies. And I forgot to tell one thing more about, like, we not only look for the Hotplate antinociception, we also look for the locomotor activity, because sometimes few compounds are kind of sedative, for example, and animal is not moving itself, and we are saying, okay, we are getting analgesic activity, but actually there may not be any analgesic activity. Animal is not moving because the compound just knocked out that particular animal. So we also look for the locomotion. I have not seen any problem with the locomotion associated with the mitragynine and 7-hydroxymitragynine. And then on the basis of this PK and PD data, we developed a PKPD model to predict what is actually happening. So the EMEX model, Michaelis-Menten EMEX model fitted the best for us. If you look at the equation, E is equal to E0, Emax concentration in the effect compartment to the power gamma upon, like, standard Michaelis-Menten reaction. And then we saw the effect of sex on the pharmacokinetics as well as pharmacodynamics of the mitragynine. So sex was added, the best fit model was observed when sex was added as a covariate for gamma. And then gamma, or the Hill factor. And the concentration produced, the 50% EC50 is here in the effect compartment. And then we have the equilibrium rate constant for the conversion of the mitragynine to 7-hydroxymitragynine. Now look at the model. How is this model predicting basically? So if we look at so first of all, we will look for the mitragynine concentration versus effect. So mitragynine concentrations in the brain is on y-axis, and logarithmic of percentage of maximum possible effect is on x-axis. So we can clearly see a linear model. The pharmacokinetic, the two compartment model was fitting best for the pharmacokinetics, and it was showing linear response between the PK and PD side. Now the question is, okay, if activity is linear to the concentration, then what is happening to 7-hydroxymitragynine? So if you have 7-hydroxymitragynine in the compartment, and then it should do something. If it is doing something, then this effect should not be linear. This effect should be governed by the concentrations of the 7-hydroxymitragynine in the brain. So let's see the same thing for the 7-hydroxymitragynine. What happened? So 7-hydroxymitragynine also showed the same thing. So instead of log for 7-hydroxymitragynine, it is like percentage MPE versus vein concentration was like absolutely linear. Okay, so we got the answer. We need at least this particular concentration to achieve any effect. So look what was the concentration of 7-hydroxymitragynine formed from the mitragynine. So when we look at those, the maximum 7-hydroxymitragynine concentration we observed after mitragynine administration is even less than the activity we saw, any activity from the 7-hydroxymitragynine dose. So the general theory makes sense. Mitragynine converts to a potent metabolite, and it should be responsible for the activity. But in practical conditions, the formed 7-hydroxymitragynine after mitragynine administration is not sufficient to do anything in terms of its analgesic activity. At the same time, that's the reason we are not seeing any problems associated with the mitragynine like we are observing with the 7-hydroxymitragynine, like all opioid related problems. So on the basis of that, I have few conclusions. So we saw male versus female effect, and we saw percentage MPE wise significantly greater in males than females. Females had lower exposure of both mitragynine and 7-hydroxymitragynine, which helps to explain the lower overall MPE results in behavioral assays. The brain concentrations of 7-hydroxymitragynine observed 4-hour administration, producing an analgesic effect of less than 10%, but still 1.4 higher than the maximum concentration of 7-hydroxy as a metabolite of mitragynine. So basically, the hypothesis which is available in the literature is that mitragynine's activity is coming from the 7-hydroxymitragynine, but in real life, it is forming, but those concentrations are not sufficient to do anything. Even those were like to achieve at least 10% effect, you need at least 1.5-fold higher than the Cmax of 7-hydroxymitragynine from the mitragynine. And then the third point is we didn't see there are strong evidence that 7-hydroxymitragynine has a negligible role in the antinociceptive effect of the mitragynine in mice. And the fourth point, the conversion of 7-hydroxymitragynine to mitragynine was not observed. So that hypothesis is also not true. And if we think about it, like how is it possible? So we have reductase enzymes, but those are available only in the lower part of our intestine, and we have not seen any of these conversion. We did in vitro studies too, like with hepatocytes, microsomes, S9 fraction, put 7-hydroxymitragynine and look for the 7-hydroxymitragynine. And we have not seen any of the 7-hydroxy formed from the mitragynine. So ultimately, I can conclude that like mitragynine converts to 7-hydroxymitragynine, the problematic alkaloid or problematic metabolite, but those concentrations are not sufficient enough to do anything, specifically in mice. We have to look if we want to do in humans, and the developed PKPD model, the PK model can be allometrically scaled and PD can be directly translated, or we are working on the PBPK model, physiological based pharmacokinetic model, to predict the human outcomes. So I would like to acknowledge the chemistry group, Dr. Christopher McCurdy, Marco, and Francisco Leon, who isolated mitragynine out of croton alkaloid, pretty pure, more than 99% pure, and then semi-synthetically synthesized 7-hydroxymitragynine. This is the Pharmacokinetics Group, Dr. Boni Ibri, Aaron Berthold, Shyam, Raju, Michelle, and Ali. Along with me, we are working on the pharmacokinetics side. And then we have Dr. McMahon's group along with Takato and Avi Patel and Nick Ho, who worked on the behavioral assays, the data I presented for the PD side. And this is a combined exercise to test this hypothesis which we tried, and we have funds from the National Institute of Drug Abuse, as well as thanks to the University of Florida, TDD-Co, and the UF College of Pharmacy. And I'm open for the questions. Feel free to ask related to this presentation or anything else.
Thank you, Abhishek, for your wonderful words of wisdom and the think tank and research experiences you shared. I would like to invite Dr. Navneet Verma, sir, for a formal vote of thanks. Sir, over to you. I think some technical issues were there. And hello, Navneet, sir, are you audible?
Yeah, he is here.
So please, you are requested to share the formal vote of thanks, please.
Sir, you are mute. Navneet, sir, you are mute.
Arun, I am audible?
Yes, you are absolutely audible.
Yeah.
Please continue.
Okay, thank you. I would like to thank our Honorable Chancellor, Sir, Shri Rajiv Kotiwal, Sir, for their support to organize this international webinar. I am also thankful to our Honorable Vice Chancellor, Professor MP Pandey, Sir, for his valuable support and guidance for the conduction of this international webinar. I am also thankful to our Registrar, Professor Sanjeev Agarwal, Sir, for their support to organize this webinar. Dr. Anu Srivastav, he is the President of Alumni Association. I am also thankful to him for their motivation to organize this seminar. And a special thanks to Dr. Abhishek Sharma for sharing his valuable experience on a topic. This topic is the pharmacokinetic and pharmacodynamic model of mitragynine and contribute of its active metabolites. Thank you, Abhishek. Thank you very much for their support to organize this webinar. And I think this type of webinar in future we will also be organized for our students of our IFTM University. I also would like to express my thanks to the Director SPS, Professor Sushil Kumar, sir, and convener of this webinar, Professor Arun Kumar Mishra, and all the member of the organizing team. Thank you very much.
Thank you, sir.
So now the time is to close this session. However, the time is always a limited factor. So I'm ending this session. And from my bottom of heart, I would like to thank each and everyone who shared their valuable time and for patience hearing. Thank you, everyone.
Okay, thank you.